Hi everyone, welcome back. So this is video number 24 in this playlist that I am currently creating, where I'm using RStudio to conduct some really basic data analysis, data science tasks. So in the previous video, we looked at the association between scale variables. So we looked at correlation coefficients, Pearson's correlation coefficient, R, in particular. In this video, I think I said at the end of the last one, what I'm going to do is talk about associations between frequency counts of categorical variables. So I'm going to talk about chi-squared analysis. Essentially, that's a very common analysis used to assess the association between categorical variables where the data is recorded as frequency counts. And with chi-squared, I'm not going to go too much into explaining the maths behind it all, but there are two common tests that are usually conducted when you use chi-squared. The first of these is what's known as a goodness of fit test, and this is usually used to see whether the frequency counts across categories on a single variable are equal, and the null hypothesis is that they are equal. So a statistically significant result is kind of evidence against this, suggesting the frequency across categories on that variable are different. So we'll see a little bit more about what that means in a second. The second common test is probably much more common than the first, really, is a chi-squared test of independence. And we use this to test whether the observed frequency counts on two cross-tabulated categorical variables. So we've got two variables. They're both frequency counts of things in categories, and what we're doing is looking to see if the frequency counts match the expected frequency. So our observed counts for those two variables match the expected frequencies. And if counts in a category of one variable are associated with a certain category of the other variable, so you've got one variable that's got two categories, and you've got another variable that's got two categories, A and B, variable one's got categories 1 and 2. If there's a strong association between category 1 on the first variable and category on the second variable, then we'll see some sort of some sort of association. We'll probably get a significant chi-square result, and that will tell us that our variables, the categories on our variables, are not independent. Okay, so I don't want to get too much into explaining the maths behind chi-squared. There's lots of other resources you can do you can look at to find that, and I've done previous videos where I've kind of gone into a bit more depth on stuff like that. But in this test of independence, this second test that I'm talking about, the null hypothesis is that there is no association between our two categorical variables, and this generates expected frequencies against which observed frequencies are tested or compared. And if the observed frequencies are similar to the expected frequencies, then the value of chi-square is going to be quite small, and the null hypothesis will be retained, so we won't get a statistically significant result. But if they're different, then the value of chi-squared will get quite large, and the null hypothesis will be rejected. And usually the expected frequencies, what we expect the frequencies in each of the cells of our cross-tabulated table, what they're expected to be is generated from the sum of the row and the sum of the column. Okay, that's all I'm going to say about that stuff. But what I will do now is I'll kind of go on and run a goodness of fit test and a test of independence. And we're using a built-in data set. So I'm going to use the tidyverse. I'm going to load that using the library function. So library tidyverse, that will be useful. And I'm going to use the iris data set, this built-in data set. Just going to make the output screen a bit bigger. Let me scroll down a bit. So I'm going data iris, and if I run that, you can see it's in the environment here. We can see it's given us the iris data set. That is a built-in data set in R, and it's got the word promise there. But if I do head iris, it's going to populate that, and there you can see we've got this data set that is 150 observations of 5 variables. And we've probably seen this before. It's a really commonly used data set in machine learning tasks for classification. So we've got 5 variables: sepal length, sepal width, petal length, petal width. So these are scale measurements on 4 variables relating to 3 different species of iris. And then the 5th variable here, species, is a categorical variable which tells us what species it is. It's either Setosa, versicolor, or virginica, I believe. But what I'm going to do is I'm going to actually do a bit of data wrangling here. I'm going to create a categorical variable using this petal length. So we've got all these different petal lengths as measurements. It's not a categorical variable, but I am going to use the mutate function and split it into 3 categories: small, medium, and large. So it'll basically split the scale for petal length into 3 equal sized groups, one capturing small measurements, one capturing medium, and one capturing large measurements. So to do that, I'm creating a new data frame, and from that I'm taking the iris data frame, I'm piping that into the mutate function, and I'm creating this new variable called size. And then I'm using the cut method. I'm saying take the petal length variable, break it into three groups, and give those 3 groups these labels: small, medium, and large. And then I'm using the select function. I'm piping all that into select, and I'm selecting the species variable and the size variable, so that I've just created here. So what we will have is a new data frame called df, consisting of just 2 categorical variables from this command here. Okay, so I'm not going to explain too much about the mutate function. I've created other videos around that, about wrangling and manipulating data and how to do those sorts of things. But you can see in my environment over here, I've got df, 150 observations of 2 variables. And what we want to do now is cross-tabulate these 2 categorical variables and see the frequency of species that fall into these small, medium, and large groups. So I can do that using just this table command, which is part of the base R function. So if I do table and then pass our df data frame object, you can see underneath here in the output window, we've got this little table, and you can see, so we've got 3 species: Setosa, versicolor, virginica, and then 3 size categories: small, medium, and large. And you can see all of the Setosa species fall into this small category. So there's 50 of them, and they're all classified as small. Versicolor, most of them are medium and a couple are large. And virginica, some of them are medium, but most of them are large. So you can see straight away we've got differences between these species and their size. So there seems to be an association between species and size, and the proportions across species and across size are not kind of roughly equally spread. Okay, so we will run a test of independence on this table shortly, but what I will do first is we'll just focus on one of these variables, that's the size variable, and we'll run a goodness of fit test. So as I said previously, we're only looking at the categories for one of our variables when we're doing a goodness of fit test, and we're asking, is there a difference in the frequency of small, medium, and large flowers? Are the proportion of all our 150 observations, are they spread equally 50, 50, 50 across those 3 groups roughly? So what I'm going to do is I'm going to create a new table just with that size variable, categorical variable we created. So I'm going to use the table command again, df $size. So I'm basically selecting just the size variable from our data frame. So if I run that, you can see down below there now in our output window just here, we've got a new table. It's just this one category, so just this one variable of size. And you can see we've got 50 small, 54 medium, and 46 large. So that's pretty, it's not exact, but they seem to be pretty evenly spread across these 3 size categories, which is worth noting. So it's kind of telling us maybe there isn't we're not going to find a significant result with this goodness of fit test. But let's run the test and see what we get. We're expecting, if these proportions were very different, then we'd expect we'd maybe get a significant result telling us that the column proportions, the category proportions are not equal, but they look fairly even to me there. But let's run the inferential test and see. So what I'm going to do here, and I've just highlighted it. So I'm using the pipe operator, which is a nice way to intuitively create syntax for your analyses. We've talked about this numerous times in this series. So I'm taking the data frame, I'm piping that into the select method or function, and I'm saying just select the size variable, and then I'm piping that into a table. So effectively what we've just done here, really, with the command above, but then I'm piping it into chi-squared.test. So it's the base R chi-squared statistical test. So if I run this, control and enter, you can see in the output window below we get a chi-squared test for given probabilities, and it says our chi-square value is 0.64, so it's quite a small value. We've got 2 degrees of freedom and a p-value of 0.7261, so it's not a statistically significant chi-square test. So what we're seeing here is, in terms we haven't got a significant goodness of fit test, so it's telling us that the column proportions are roughly equal, which we kind of suspected. We suspected, and we knew it was likely the case, because there's roughly about 50, there's a little bit of difference there, but there's roughly about 50 observations in each of those. You know, there's not one where there's kind of 100 in one and almost 25 in another and 25 in another, you know. So we've got a non-significant test suggesting that there is a good fit across those proportions, essentially. Okay, so that non-significant result, as I said, gives us this indication that the counts in each category are approximately equal. One thing I was going to demonstrate though is here, if you look here, I've got alternative syntax if you prefer nested statements. So I've used the pipe command to run this test here. An alternative way to run exactly the same command, which is, you know, it depends on which you prefer, but you could think maybe this is a little bit more succinct. Is you can see I've got the chi-square.test, chi-sq.test, and embedded within that, nested within that, I've got a table, and I've selected the size variable from our data frame. This will give us exactly the same output as the test that we ran above using the pipe operator. So let me run that, and you can see, there you go, we get exactly the same result. Okay, so that was goodness of fit. So we actually had a good fit across our columns, across our categories, across the levels of that categorical variable size. Okay, so not that commonly used, I don't think, but much more commonly used when you're doing chi-square test is the test of independence. And that tests a null hypothesis that there's no association between the categories across two variables. If the test is significant, then the variables are not independent, and knowing the category of one independent variable tells you something about a category on the other variable. Okay, so is there an association between the counts in one category relative to the other? So we're going to go back to that original table that we put up where we cross-tabulated both of our variables, and you can see there we've got it back. We've got our species in the rows and our size in the columns. All of the Setosa are in this small category. Most of the versicolor are in this medium category, and most of the Virginica are in this large category. Okay, so I'm gonna use I'll demonstrate both types of syntax using the pipe and using nested syntax. So to do this, pretty simple. We just take our data frame, pipe that into the table, then pipe that into the chi-square test. And there you can see this time we've got a chi-square value of 256.52. 4 degrees of freedom, and a p-value is less than 2.2 times e to the minus 16. So that's a very, very small value. So we'd have to use this, move this decimal place like 16 places to the left to get, so it'd be 0.000 and carrying on until we got to like the 16th digit after the decimal. So on this occasion, we have got a significant chi-square test. So this is telling us that the categories, the levels of our two categorical variables are not independent. And we kind of knew that because we knew that size and species were related in some way just from looking at that data. That's kind of the reason I chose this particular variable. It kind of illustrates it quite well. Okay, so I'll demonstrate the alternative syntax as well. So using this nested type of syntax where we've nested table and selecting the df within the chi-square test gives us exactly the same output as before. So we've got this significant result. The variables are not independent, and knowing something about the size of the species tells us something about which species it is likely to be, and vice versa. But what we've got here is minimal output for a chi-square test, and kind of the minimal thing you'd be interested in, the test statistic, the degrees of freedom, the p-value. But often you're going to want to report more things than that when you conduct a test like that. So you're going to want to know what the observed frequencies are, what the expected frequencies are. So what I'm going to do is I'm going to run that analysis again. I'll use the nested syntax here and save it as an object called res for result, essentially. Let me run that. It hasn't printed any output because I've not called res. If I kind of typed res below here like this, it would, and ran that command, it would print the chi-square output that we saw above. So this Pearson's chi-square test. But what we have with this particular object is a number of attributes that we can call. So if I use this attributes function and pass this new res object, if you see here in the environment window, it says res, the result, it's a list of 9, so it's a list of 9 things essentially. If I call attributes and then pass in our res object, it'll tell us what those 9 things are. And there you go, you can see we've got a list of names, different things that are part of the analysis we've run. So we've got a statistic, parameter, the p-value, method, data.name, observed, expected residuals, and standardized residuals. So those are kind of useful bits of information. So if we run, we can select these individually and print them out. So if we run res then $expected, so ask for the expected attribute for our result, you can see it gives us this table showing what the expected counts were. So these are the counts that it expected to see under the null hypothesis, that there is no association, there is no relationship between species and size. And you can see they're all, so we've got 16.66 in all rows for small, 18.15, 18 in all rows for medium, 15.33 in all rows for large. Okay, so an important thing to note with regards to chi-square tests is if you have a small data set and you just cross-tabulate in a 2x2, so one variable with 2 categories, another variable with 2 categories, you need to you can get erroneous results if more than 20% of your cells, which would be each of these intersections between 2 levels, if more than 20% of your cells has an expected frequency of less than 5. We're okay here because there's 16, 18, 15, they're all above 5. But usually if you have that, when you run the test, R will print out a warning saying that the results could be wrong because you're kind of not meeting some of the necessary requirements to run that test. And if you did have that, then you'd need to do a different type of test, which would be Fisher's exact test. I'm not going to run that here because I don't want to spend too much time on chi-square. Really, I don't use these types of analyses that often. But often you collect data, you do research where your variables are frequency counts of something. So these types of methods are worth knowing and worth understanding. Okay, so let's have a look at some more attributes before I finish. So we can get the observed counts. This is going to be just the tabulation of our observed data, which we've already seen. Another thing we can ask for is the method. If I press that, you'll see what that gives us. It tells us what test was run. So Pearson's chi-squared test. And the last one I'll do is we'll ask for the statistic attribute. And this will just print the chi-squared value from our test to the screen. And you can see there it was 256, so a very large chi-squared value, because we saw what was, just looking at the table, an obvious relationship between species and size. And we confirm that with this inferential statistical test, that there is these variables are not independent. There is an association between them. Okay, I think this is long enough, and that's enough time to spend on the chi-square test. I'll have to have a think about what the next video is going to be. Maybe we'll move on to the t-test. Maybe I'll start looking at linear regression, something like that. But thank you for watching and listening. Hopefully this was useful, and hopefully now it's kind of, if you're kind of playing around trying to run these types of testing, or this is helpful, and you will be able to use this type of script and do that. Okay, thanks very much, and I will speak to you soon.